This subject dropped into my lap probably about 4 years ago, and I'll tell you what a change it's made in my life. I've been working 7/24 and has been on like a discovery high for 4 years. It's unbelievable where this field has gone. And the understanding of chronic disease is actually, in the last 5 years, made more progress than you can imagine. It's the things we're learning about the root cause of chronic disease, and that is probably the most important discoveries we've made in the history of medicine, in my opinion. And I would say within the next 10 or 15 or 20 years, there's not going to be any more chronic disease. Cancer is going to be very rare. All these things that we're suffering with in the last 2 decades of our lives will not be there. They'll be very rare because of things that we're beginning to understand about this whole subject. So I've only got 30 minutes, and it's a 2-hour lecture. So I'm going to do in 30 minutes what that is 2 hours, and try to give you enough to stimulate you to go look at the lecture, and also I have 2 books on the subject. But chronic disease is a worldwide epidemic, and it's been in the last 100 years, and by the time you hit 60, 80% of us have it. So what in the world has changed in the modern world? You know, and then how, of course, I'm a dentist, implant dentist, and because of my experience in implant dentistry, I was able to find out why so many of my patients with missing teeth actually had these chronic diseases. So I think MS is just, in my book, is just another chronic disease. So I look at the root cause being the same as what I'm going to talk about here. So when you hear me talking about chronic disease, you can lump MS along with it. And I'm thinking that the bacteremias from periodontal disease, which we know we have every time we brush our teeth or chew food, if our gums are all boggy and bleeding, we get bacteremias of the bugs that get from the mouth into the bloodstream. And they dump through the facial artery into the jugular vein. And from what I saw today, I just added this, that backflow carries that bacteria right to the brain. So I just added that to just an hour or so ago. So it's something the researchers can look at and see if that actually is true. The World Health Organization calls these non-communicable diseases, and it's a worldwide epidemic. But in reality, they're not. They're not non-communicable, they're communicable. And the more you study the root cause of chronic disease, the more communicable it becomes. And I'm going to talk about spirochetes and spirochetes getting out of whack. Well, the cause of the chronic disease is the underlying cause, or the immediate cause, I feel, is the bacteremias of these spirochetes, and then you end up with a syphilis-like long-term chronic inflammatory disease similar to syphilis and Lyme disease, and I call it oral spirochetosis. But what made those spirochetes get out of whack? That's and it's the terrain. And there's a couple things that I'm going to mention very briefly, but I've got a blog. It's called lifeguardyourhealth.com, and you can go there and get more information on this because I keep adding more and more to it. So I'm not going to be able to get through the whole thing, and so you'll be able to get more information from that. But Weston Price, many, many years ago in the 30s and the 40s, went around to all the indigenous populations all the way around the world, and he studied people's whether they had cavities or not, and also diseases. And every population of these indigenous peoples all around the world back 50, 60, 70 years ago, they didn't have cavities, they didn't have chronic disease, they didn't have crooked teeth. But when these people moved to populated areas and got on modern diets, and then they went up to 80% cavities, and within a generation or two, they got malocclusions and bad teeth and started getting diseases. So we've known about the diet and its causes for many, many years, but in dentistry, we've denied it and say, oh, that's a bunch of bunk. But now we're coming back to it after all these years, and that actually the terrain that's caused by our modern diet skews the bacteria flora toward more pathogenic bacteria. I've got African American patients that have diabetes and heart disease, and they say, well, my relatives back in Africa, they don't. Well, that's true. My co-author in my last book, Dr. Krushkos, he's a professor emeritus, University of Connecticut. He's an oral pathologist, and I wrote our last book called The Silent Saboteurs. But we're just publishing a new paper in a journal, Connecticut Dental Journal, and it's coming out next month, and it's on a concept of what's called the metagenome. The DNA within our body, the human DNA represents only 20% of the DNA that's represented in our body. The 80% is bacterial, virus, and fungus. And so essentially what we have in our body is a microbial garden, and that garden needs to be attended. And so what influences that microbial garden, specifically sugars, refined carbohydrates, corn syrups, because that bacteria likes it better than you do. And these modern diets are very, very addictive. When you get on these modern diets and fast foods, it's extremely difficult for patients to get off that and get onto a healthy diet, which is essentially eat like a gorilla, but not as much. And then your salad should be your main meal, and everything should, the refined carbohydrate, sugar, and that should, if you want to get over these chronic diseases, number one thing is get off sugar, get off refined carbohydrates and corn syrup today. I couldn't emphasize that anymore, because it influences the microbial garden toward more pathogenic bacteria. And the thing of it is, what caused the problem? We talk about dental metals and all that. Well, what caused the need for those? Well, it's the same thing. You know, this diet caused the cavities, or it caused abscess teeth. And we now know that root canal teeth are the same bacteria that we're going to talk about with gum disease. So the root cause goes way before the metals were even put in the mouth. So we need to get to the root cause, which essentially is diet. It's things we already know. So in this lecture, I lectured to dentists, and I took out the whole section before here as failing implants. And the whole thing with failing implants we find with people with a lot of missing teeth is a depressed immune system from the things we're going to talk about. And so with a depressed immune system, it's very hard for people to fight various things, and especially infection. So essentially, chronic disease, if you think of chronic disease as a big open sore in the mouth, gum disease, think about an open sore as big as your palm of your hand. When you have full-fledged gum disease, that's how much open wound you have in your mouth. And in the mouth, there's probably 500 different types of bacteria, many of which are extremely virulent, but they're in small numbers. So when gum disease gets to the point where you have bone loss, the bone loss find is related to the chronic diseases. And spirochetes are 80% of those bacteria when this happens. So when the spirochetes get in your system, the phagocytes come, and then it just turns into an offensive defensive battle, which there's no way I would have time to explain how that works, but it's chronic disease is like a stalemate between the immune system and the bacteria, especially certain bacteria. And it's a never-ending immune response as a result. And you're going to have to do the research, read my books, and get into this, because there's no way that I can. One patient came in, and I've been studying this for many years, and this is actually a phase contrast microscope that I rigged up with a 18 megapixel camera, high definition, so I can get a macrophage this big and watch everything live. But this happens to be a patient who doesn't have any periodontal disease, but had an abscess tooth that was draining. So I took samples, and I could see what was going on in this situation. And what you see is little vacuoles or vesicles, but also spirochetes mimic these particular structures. And I'll show you that later. I'm going to show you the difference between a normal acting immune system and a suppressed immune system. And here, if you look at these are all immune cells here. They're all intact. And within them, you see a lot of movement. It's bacteria. They seem to be like little lobster traps. They don't like go phagocytize them like that. At least the bacteria seems to be attracted to them, and they go in them. And when they go in, they can't get out. So that's the normal acting immune system. And here is a suppressed immune system, and I've been looking at this for almost 15 years on patients. We take a plaque sample out of the mouth, put it on a high contrast microscope, and this happens to be a trichonomad that you find these, and this happens to be an Alzheimer's patient. And you find lots of spirochetes, trichonomads, you see now I'm blocking that, amoeba. And here is a macrophage right in the middle. And you see little spirochetes on the side. I don't have a pointer right here. But the macrophage itself has nothing inside it. You don't see any bugs at all. But you see spirochetes trying to drill holes in it. You wonder who's winning the battle here. And this is a suppressed immune system. And you can find this on anybody that has periodontal disease, that the macrophages don't work. And the immune system doesn't work. And the bacteria is that's happy as can be all around the immune cells, but nothing's going on. So in my opinion, when you study spirochetes, there's plenty of studies and animal studies that show that, like if you put spirochetes into an experimental animal and seal it off, eventually you'll find an abscess, and then when you sacrifice the animal, those spirochetes will be everywhere in the body. And there's no other bacteria that they studied that could do that. So it seems that spirochetes, like syphilis and Lyme disease, have the ability to go anywhere in the body, and once they get to where they're going, they transform into these what we call pleomorphic forms, set up the immune response. Once the immune response is set up, then we can get co-infections similar to Lyme disease, and I think periodontal disease is the same thing. But once those pathways set up, then this battle goes on, and it can occur anywhere in your body, and depending on where it is the name of the disease. There's probably 300 or 400 named diseases, all caused by the same thing. So I got onto this subject because I was studying why all my patients with a lot of missing teeth all had diabetes, heart disease, various degrees of dementia or diminished cognition, and so I thought, what is going on here? All my patients that I do a lot of implants on are sick. So this is what started me, and I lectured on this for many years before I figured this thing all out. There's a very large Japanese study, and we've been waiting for a study like this because us in dentistry that do dentures know that a lot of those denture patients are like, a lot of missing teeth, a lot of missing screws. That's just the way it is. And they're really hard to work on, and it's not the denture. Dentures are easy to make. It's the problem with the cognition part of these patients. And in implant dentistry, we have a very big problem with lawsuits. And the reason is the same type of people that, have a lot of missing teeth, they have health histories of chronic diseases and then cognition problems, and they don't understand what you're talking about. They don't understand if things go wrong, what you can't explain to them, and it's a cognition problem. So now we know from Judith Micklowski that she just came out with a paper probably 6 or 8 months ago, and using Koch and Hill's postulates, has come up with the fact that Alzheimer's disease is actually caused primarily by oral spirochetes. And here's Micklowski's work in showing, first she did all spirochetes, and that's the front part is controls and the back part is what they found in the brain of all spirochetes, and then the second one is oral spirochetes. So in Alzheimer's disease, they're finding out that they're finding spirochete DNA in over 90% of cases. So, and that's only looking at the helical form, and they didn't look at some of the other pleomorphic forms. Depression is related to periodontal disease. I'll go through arthritis. Chronic disease, inflammatory disease related to periodontal disease. There's just so much references in the literature today. The Surgeon General came out in 2000 with the same thing. And the cost is just overwhelming our medical system today. I mean, it's just unbelievable. Trevor Marshall is a very important researcher and has helped us understand this chronic disease thing. And he talks about comorbidities. And you take all these names all the way around this circle, all different chronic inflammatory diseases. And we wanted to see if you had like periodontal disease, what other chronic disease you have. And so he went and took each one and went through PubMed and through his research and connected the dots. So when you get all the way around the circle, I mean, all these chronic diseases are all related to each other. And it's quite amazing. If you take normal saliva and check the DNA, there's 500 different bacteria, and if you look in there, but some of them are very bad actors in bacteria. Actually, Treponema palatin, the spirochete that causes syphilis, is one of them. And a lot of us didn't know that. This is, by the way, a plaque sample with people that have a lot of spirochetes, and you can see and I'm not going to belong this one, but when you're looking at plaque samples of what we call a normal microbial garden, you don't see any activity at all. And when we're treating patients with periodontal disease, we use a microscope, and this is what we're shooting for. Nothing moving around, and when we get to this point, this is what we call a normal microbial garden in the mouth. If you get a chance to get this lecture, it talks about how he discovered all this, and the doctors that treat atherosclerotic heart disease think there's an injurious agent. They don't know what it is, and I pretty much showed what it is. I want to show you this video because it really is apropos, because in atherosclerosis we now know that those plaques they're loaded with DNA of bacteria, especially spirochetes.
But instead of making an X-ray image of the center of the artery, Nissen was going to try to use a miniaturized ultrasound probe to scan the cells that line the artery wall. There was just one problem.
At the time we were doing this development, the smallest ultrasound devices were about the size of your fist. And we thought we could make an ultrasound device so small that we could actually put it inside the coronary arteries. Now, coronary arteries are very, very tiny. They're about the size of a pencil lead. And so like a radar dish goes around, it rotates around, sending and receiving these sound waves, just like sonar. And when it sees the reflections come back, it makes a picture from those reflections. I will tell you, it was very frustrating. A lot of the work was done in animals because, of course, you can't test something like that in humans until it's been perfected. And we would go into the animal lab, and we would not get a picture. And we would say, well, all right, let's go back to the drawing board. But by about 1989 or 1990, we were making pictures.
I'm going to go ahead and put this catheter inside the artery, and then I will be able to move back and forth to look at every micrometer of the coronary artery wall.
We put one of these catheters in the coronary artery, and there was all that plaque that we couldn't see any other way. You could see on a television screen, just as if you were literally there. And it really was like a fantastic voyage. It was a trip inside the arteries.
The new technology called intravascular ultrasound, or IVUS, was working, and the Cleveland team was shocked by what it revealed.
So here's an example of how much you miss if you just look at the old-fashioned way at the angiogram. And this is an angiogram. You don't have to be a cardiologist to understand it. So you see this is the dye in the coronary, and you see everything's nice and smooth. Nothing is actually narrowed. So now we're going to take you inside that artery, and we're going to show you what the ultrasound looks like. Now remember that the ultrasound is going to look at the artery in cross-section, like you took a sausage and sliced it up. And there's a gigantic plaque, which I've colored in.
Okay, it goes on a little bit longer. What this shows that with this new technique, they were seeing 99% more lesions than they did with the old angiograms. And so my point is that I know that they're not looking at this as far as the veins are concerned, as to the constrictions caused by the same thing that cause atherosclerotic heart lesions and these lesions in the arteries. So, and I've talked to a couple of the doctors about this, and I think that they need to take a look at this. And what we're finding is that biofilm that's actually in that lesion is the same type of biofilm you find in the mouth. And I don't know how much more. I want to see if I can get to one more thing on this. I related oral spirochetosis to Lyme disease and syphilis, and that's what this is about, and you can get this if you want. And I actually took that same chart that Marshall had and put periodontal disease and went through my charts to see what comorbidities we had with periodontal disease. And you can see, I mean, it's almost every one of those diseases was represented when I was checking my own charts, patients with multiple missing teeth, and missing teeth is a proxy for periodontal disease. Now, we didn't talk too much about spirochetes, but let's see if I can get. Now, spirochetes are preamorphic, and here is a paper that goes back to the turn of the century, and it shows how at the end of life, over here, they start forming these little granules. And those, they sell about 16 or 18 of them, and when the cell membrane breaks down, they go into the plasma and the fluid around them, and it produce spores, each of which can produce another spirochete. And this is Lyme disease, and we find they produce these cyst forms as well. And what I think spirochetes do is all they want to do is just get to the end product of a cyst and sit there and be dormant, even though they still produce an inflammatory reaction, and wait for the next host. And when that person becomes a cadaver, I mean, humans aren't eaten by others, but probably at one time in history they were. But in the animal world, then they become cadaver, which completes the life cycle. So it goes from cadaver to chewing, getting your teeth, and the incubator is periodontal disease, and then they get in your body, go to the end point, which is the muscle, and then cadaver, and that's, or carrion, and that's your life cycle of a spirochete. Now, what I'm showing here is right within a phagocyte, there's a spirochete being phagocytized, and actually show how these granular forms are actually being formed. So phagocytosis really doesn't kill a spirochete. Now here's what happens when you treat a spirochete with penicillin.
Inside a slide, and these are from pure culture from Borrelia burgdorferi, and you can see objects flying by, but he's got a wicking of a very dilute solution of penicillin going through, and he's watching these spirochetes as they react to the penicillin. And
They
so
zoom.
they roll up into that little spore-like
These
form
things
that
mimic-
he calls a cyst.
These things mimic what I showed you right at the little vesicles that are formed in phagocytes. And it's if you were going to design an organism or like a bioweapon and then design a spirochete, and then it hides and mimics another structure that occurs in immune cells and also in epithelial cells, these little vesicles or vacuoles, then these spirochetes mimic them almost perfectly. So it's just amazing. And here's electron micrograph of one that goes back into 1948 when electron microscopy just came around. And here's the mechanism from Wolf from Germany that shows the progression of a spirochete to these little spore-like forms. Here's more of this is from Chang from Montreal, then showing the same thing. And here they are again. And this is a very recent study, this 2011. And this one is taken from epithelial cells and is from the Journal of Dental Research. And they suggest the fact that because the epithelial cells in the lining of gingival sulcus, if they're not, if these spore-like forms that are actually in the cells themselves aren't removed, that they repopulate the gingival sulcus and periodontal disease. And one more, and then I'm quitting, okay? This is immunofluorescence, again, a very recent, well, it's 1995. And this is showing one of the circular forms here. So we know that spirochetes actually form these pleomorphic things. And when you're taking plaque samples, you can actually see the epithelial cells with these little spore-like forms in them. You can find in the atherosclerotic lesions, you find these same structures. So we know that atherosclerosis is actually caused, for the most part, by an infection. And so I wouldn't be a bit surprised if we find the same thing in the venous part in the jugular vein as the research progresses. I think I got my point across, I hope. And if I didn't, there's plenty of stuff you can get online that you can get a hold of.